Hello and welcome. Today I am going to take human genetic disorders. So the topic is human genetic disorders. So we have human genetic disorders. So basically, soon after the rediscovery of Mendel's laws of inheritance, a number of disorders in human population were found to be inherited. It's actually Archibald Garrod in 1902 and William Bateson 1902. They followed the laws of heredity. The disorders have also gave insight as to how a gene controls metabolism and phenotype. Genetic control counseling based on the pedigree analysis is important to check and separate the genetic disorder. So, if you see there are a lot of metabolic and some genetic disorders. So basically, we're dealing with the present topic. It deals with human genetic disorders. So these are actually gene-based disorders, but likely they affect everything of the cells, chromosomes, etc. So. Archibald Garrod and William Bateson in 1902, they had a lot of study on this. So first of such disorder which we are discussing is the hemophilia. So first is hemophilia. Okay, so this hemophilia actually it's called a biliary disease given by John Otto. So he was actually John Otto in year 1802. It's a biliary disease. It is sex-linked recessive trait. So it's first of all it's a sex-linked. So that means it's present on sex chromosome, and it's a recessive trait. That means it is not shown as such until it's expressed because of lack of dominance gene. So it is a sex-linked recessive trait which is known as biliary disease because the exposed blood does not readily clot due to deficiency of plasma thromboplastin or factor 9th, this hemophilia B, this Christmas disease, or it can be due to anti-hemophilic globulin, that's factor 8th, that is hemophilia A. The defect has been inherited in family of British Crown through Queen Victoria. So in females, hemophilia appears when both the sex chromosomes carry this recessive gene. Such females die before birth. A woman having a single allele of trait is normal but is a carrier of the disease. For sex-linked genes, humans, males are homozygous. Therefore, if we take X, H, and Y, this person is hemophilic. Now, we have a lot of crosses between different criteria, different man, woman, and we will get different kind of offsprings. So second is we're taking is red-green color blindness. Red-green color blindness. So we have red-green color blindness, or it's simply called the color blindness, or it's called daltonism. Okay, now it was actually Horner, Horner and Wilson in year 1876, and then Wilson in 1910, who have a lot of study on this. Now it is a recessive sex-linked defect in human beings, because human beings are able to distinguish between red and green color. Gene complement of the color blind woman is definitely, that's XC, and that of color blind man is XC and Y. So and career woman, the career woman having normal vision can be X, this one. Now marriage between different persons will give different results based on this typical phenomena. And color blindness is basically of three types. We have protanopia, that is red color blindness. So if we take protanopia is a red color blindness. We have deuteranopia, that is green color blindness. We have tritanopia, that is blue color blindness. We have third genetic disorder, that is sickle cell anemia, that is sickle cell anemia. So if we take anemia as basically this is a low content of RBCs. Now sickle cell means these RBCs get kind of sickle shaped sieve structure. So it is due to codominant formerly considered recessive, but it's autosomal allele HBS and present chromosome number 11. So we have HBS individuals die during infancy, but HBA, that means who is having a good than HBS, so these individuals survive. The allele has survival value because heterozygotes have an advantage over normal individuals in malaria infested areas because they resist damage from Plasmodium falciparum, because 30% population carries the allele in Nigeria. So this is sickle cell, he dies. This is the heterozygote sickle cell and normal. This one, it resists this malaria. So these people have that advantage to resist malaria. Now, because then that of a normal one, okay, we have HBS. HBS produces hemoglobin with, that means this sickle cell. If we take the sickle cell hemoglobin over this cell, then HBS, it produces hemoglobin with beta chain. So HBS, it produces hemoglobin with beta chain. We have alpha and beta chain hemoglobin because there's a protein, the secondary structure protein. So beta chain having valine amino acid. So we have valine amino acid instead of glutamic acid. So instead of glutamic acid, that is the difference. So this is having valine instead of glutamic acid at 6th position. So at 6th position, the hemoglobin beta chain, the chain is beta chain, position is 6th. Glutamine is replaced by valine. This is all given by actually Ingram. That's 1958. So heterozygotes are normal individuals with 60% HbA, 35% and 40% HbS. They develop sickle cell crisis under conditions of oxygen distress like altitude and strenuous exercise. At this time, 6-valine develops hydrophobic bonds with complementary site of an adjacent citrate. Other interactions amongst molecules also occur, causing development of helical polymer of about 14 citrate. Okay, it changes membrane permeability and causes development of sickle cell form. Sickle cell cells are rigid and obstruct capillary blood flow. It results in further deoxygenation and sickling. A mild form of sickle cell anemia occurs in Sinhalese. So they have mild kind of sickle cell anemia. That means, so because of all of this change, the 6th position of a glutamic, this beta chain, as compared to normal, gets some kind of hydrophobic interactions, changes. Finally, cell becomes sickle shaped. It is not a normal cell. So this is sickle cell anemia. Now we have the fourth one is Phenylketonuria. So we have fourth genetic disorder, Phenylketonuria. Do you know what is this? It's shortly called PKU. It is homozygous recessive autosomal alleles present on chromosome 12 cause absence of phenylalanine hydroxylase in liver. So that means homozygous recessive autosomal allele that it involves chromosome number 12. Here we have a problem. There's absence of phenylalanine hydroxylase enzyme is absent in liver. The enzyme essential to change phenylalanine to tyrosine. Because enzyme is absent, the phenylalanine gets on to accumulated. There is increase in phenylalanine in blood, hyperphenylalanemia, and also in urine. Byproducts of the phenylalanine also occur in excess. They include phenylpyruvate, phenylactate, phenylacetate, phenylacetylglutamine. There is severe mental retardation, hypopigmentation of skin, and hair, eczema, mousy odor of skin, hair, and urine. That means it is basically the deficiency of enzyme because of deficient gene because of genetic defect with regard to production of that enzyme. Finally, taking the fifth one, we have cystic fibrosis. Cystic fibrosis. Now, what is cystic fibrosis? It is again a recessive autosomal disorder due to alleles present on chromosome 7. So, there is a failure of chloride ion transport mechanism. So chloride ion transport mechanism fails. Elevated levels of sodium chlorine ions occur in sweat, thick mucus collectors in lung and airways, leading to blockage and secondary infection. And antibiotics and physiotherapy have increased average life of patients from 5 years in 55 to 25 years in 1995. The disorder was formerly called mucoviscoitis. So it was called mucoviscoitis formerly. Okay, so cardiac failure may occur. In most cases of cystic fibrosis, there is an impairment of pancreatic and liver function. That is common in Caucasian population. Okay, now let's take the sixth one. So we have a sixth one, and that is Huntington's disease or Huntington-chorea. So it is Huntington's disease or Huntington-chorea. So that's a different kind. We have, it is dominant autosomal disorder. It is not recessive. It is a dominant. So whenever it is present here. It is a dominant autosomal disorder due to an allele on short arm of chromosome 4. So chromosome 4 has a short arm, has a defect. It produces an inhibitor of brain cell metabolism. The characteristics of this may start at the age of 15 to 40 years, but can occur in still older persons. There is atrophy of parts of brain resulting in high respiratory irregularity, faulty articulation of speech, irregular arrhythmic movements of the limbs, a peculiar dancing gait, and bizarre grimacing. So Huntington-chorea is such kind of a disease. It's not so little, it's a big problem, we can take one more. That's overall we have some other recessive autosomal disorders, other recessive autosomal disorders, red. So recessive autosomal disorders, they are exactly, if we take first one, there is thalassemia. Now what is this one? It's a group of actually genetic disorders which result from defective synthesis of subunits of hemoglobin. In alpha thalassemia, out of 4 genes present on chromosome number 11th, absence of 2 genes, that is alpha, and or that's alpha dash. Now what happens here is that it produces microcytic and slight hypochromic erythrocytes without significant anemia, but deficiency of 3 genes, that's dash alpha dash, and it develops microcytic hypochromic erythrocytes with marked hemolytic tendency. Now death occurs in fetus in case of deficiency of all the genes. In beta thalassemia, so we have beta type also, the 2 beta genes present on chromosome number 16th chromosome are defective. In beta thalassemia minor, 1 defective B gene, the erythrocytes are microcytic with higher RBC count but 15% lower hemoglobin than normal. In beta thalassemia minor, so this is a minor and this is a major. We have 2 types of beta thalassemia. So first of all we have thalassemia alpha beta, then we have beta the minor and major. In beta thalassemia major, Both the genes are defective. Disease manifestation begins at the age of 4 to 6 months, when switches from this gamma chain to beta chain. So during maturation of RBCs, there is a severe anemia with microcytic hyperchromic RBCs. So this is a problem with this thalassemia. We have second, this recessive autosomal disorder, second RAD, and that is Alkaptonuria. So we have Alkaptonuria. Again, homozygous recessive, they do not produce liver enzyme hemogenetisate or alkapton oxidase. So it is a problem of enzyme. So homogenetic acid, homogenetisate enzyme, or we can say alkapton oxidase is not produced, which is essential for metabolism of hemogenetic acid, or which is called alkapton, formed from phenylalanine and tyrosine. As a result, alkapton accumulates in body, producing arthritis and other damages. Alkapton is also excreted in urine. Exposed urine appears brownish black due to oxidation of hemogenetic acid. This is a problem. So black urine comes out. We have third one of it, third RAD, that is albinism. Albinism. Now, do you know what is this? It is actually enzyme tyrosinase. It is also inhibited by excessive phenylalanine. Enzyme tyrosinase is absent due to homozygous recessive autosomal allele. Melanin or pigment formation from dihydrophenylalanine is stopped. There is a lack of pigment in the skin, hair and iris. So in skin, hair and iris, the pigment is not produced. Because of this, eyes disorder may occur due to damage from bright light. Albinism is most commonly due to recessive allele on long arm of chromosome 11. But can that means also be caused by another recessive allele of P gene on long arm of chromosome 15. So both chromosomes get involved in albinism. Now we have one more, that's Tay-Sachs disease. Tay-Sachs disease is also called infantile hemorrhagic idiocy. Infantile hemorrhagic idiocy. So it's actually Tay and Sachs 1881 and Sachs 1887. It is a recessive autosomal disorder which appears after birth due to deficiency of enzyme beta D and acetyl hexosaminidase. So this enzyme beta D and acetyl hexosaminidase, there's a problem in it. It's a deficient actually. It causes accumulation of lipid GM2 or Tay-Sachs ganglioside. So it's actually metabolism related problem. Tay-Sachs ganglioside GM2, it gets accumulated, that damages brain and spinal cord, resulting in mental retardation and paralysis. Death of child occurs at the age of 3 to 4 years. Now, very bad luck for those people having this 3 to 4 years child between this age gets died. Now we have one more, that's in fact fifth one, and that is muscular dystrophy. Muscular dystrophy. Now this disease, 3 types of muscular dystrophy occur due to recessive autosomal alleles, that is limbic, limb girdle, distal and congenital. 3 types of muscular disorders occur. Now we have, these were 7. Now we have 8th type. We have 8th type. Now if we take the 8th type, and that's other dominant autosomal disorders. So these are other dominant autosomal disorders. So those were Recessive autosomal disorders. Rad, this is dad, dominant autosomal disorders. First of all, we have polydactyly. Polydactyly. So that means having more than 5 digits. Having more than 5 digits. One more is that is called the achondroplasia. You know, we have achondroplasia. It is an inherited dominant autosomal disorder where the long bones do not grow, resulting in dwarfism. Intellect is not affected. We have one more, that is muscular dystrophy. So we have this muscular dystrophy, but it is a recessive one. This is dominant one. Muscular dystrophy due to dominant autosomal gene can be of 4 types. This had 3 types. This is having 4 types: myotonic, fascioscapulohumeral, oculopharyngeal, and scapulo that is peroneal. 3 types this muscular dystrophy we have. Taking the ninth one, that's other sex-linked disorders. Now we have genetic problem. These are sex-linked. So these were autosomal. Now we have sex-linked disorders. First of all, we have glucose 6-PD deficiency syndrome. Glucose 6-PD deficiency syndrome, the sex-linked recessive trait present on chromosome X, homozygous in females, single in males, causes deficiency of glucose 6-phosphate dehydrogenase, which is essential for carrying out hexose monophosphate shunt. The shunt is required to produce reduced glutathione for protecting sulfhydryl groups of hemoglobin and erythrocyte membrane during oxidative stress. In the absence of GDP, hemoglobin crystallizes and erythrocyte membrane ruptures. Beans, hence Favism, it's then called beans, and certain drugs induce hemolysis in GDP deficiency persons. So we have beans and some drugs, they cause hemolysis. So because of beans, this can be called Favism. Okay. Now vitamin K, chloramphenicol, and quinine, no effect on certain. They have effect p-aminosalicylic acid and phenacetin, actinolide, sulfonamide, sulfonamide, chloroquine, primaquine, etc. have these. These are drugs. These are problems. Now this was glucose 6-PD deficiency syndrome. We have one more of this sex-linked is congenital night blindness. We have congenital night blindness. The sex-linked recessive trait causes reduced development of visible purple and hence night blindness. Congenital night blindness is more prevalent in males due to presence of only one X chromosome and hence expression of even a single recessive allele. Night blindness caused by vitamin A deficiencies, acute night blindness. Okay, these were 2. Now we have third one, and that's muscular dystrophy. Again, we have muscular dystrophy. So we have a lot of number of muscular. So this type of muscular dystrophy, is actually is a sex-linked muscular dystrophy, and it has basically 2 types. One is Duchenne's pseudo-hypertrophic muscular dystrophy. So it is Duchenne's the pseudo-hypertrophic muscular dystrophy. It is which is onset at the age of 5, with progressive weakness of girdle muscles, inability to walk after the age of 12, cardiomyopathy, mental impairment, respiratory failure in second and third decade. The dystrophy appears due to the non-synthesis of protein dystrophin. The protein is required for the transfer of nerve impulse to calcium storing region of the muscle. Muscle contraction is poor here, and excessive calcium also depresses anti-myosinic level. So we have this one. This is Becker's or benign pseudo-hypertrophic muscular dystrophy. This is second type. This is actually mild form in which symptoms appear late and are less severe. So respiratory failure occurs after the age of fourth decade, and this is the second type. So this muscular dystrophy is two types. Now we have 10th, and that is Alzheimer's disease. So let us talk it here. This is 10th, this Alzheimer's disease. So let's write Alzheimer's disease. It's a progressive degenerative disease of brain, which is caused by accumulation of amyloid senile plaques inside nerves, resulting in their degeneration. There is a development of neurofibrillary intangibles resulting in loss of choline acetyl transferase activity. So it's amyloid senile plaque found inside. The disease is believed to be caused by two defective alleles, one on chromosome 21. So we have problem on chromosome number 21 and the other on chromosome number 19. So we have 21 and 19. As chromosome 21 is also involved in Down syndrome, that means Alzheimer's disease can also occur in these patients. Here Alzheimer's disease occurs early in Down syndrome patients. More common form of disease is associated with the allele present on the chromosome 19. So it produces disease after the age of 40. So you can say that the disease results in we can say allele present on it results in non-metabolism of amyloid beta peptide. So it is amyloid beta peptide actually. And first signs are subtle changes in personality, memory disturbance, and trimming of hands. It is probably progressive increment in increase in dementia over next 5 to 10 years. So there is a loss of memory after that. So that's a profound symptom. So we have 11th one. This is autosomal abnormalities. So this time we have autosomal abnormalities. There is a large scale of possibility of autosomal aneuploidy in human beings. However, very few are known, as most of the aneuploidies result in spontaneous abortions. So in fact we have three of them. There is three important ones can be that's Down syndrome and others. So first of all we write Down syndrome or Mongolian idiocy or Mongolism. It was L. Down in 1866 actually. So he cytological bases were given by J. Legioni in 1959. So it's due to trisomy of 21st chromosome, that is 47 plus 21st. The syndrome is characterized by rounded face. So face is round. We have broad forehead. Then we have small ears. So ears are very small. We have flattened nasal bridge. We have open mouth. We have projecting lip. Then so we have this the lip, lower lip is projecting, this lower lip. Then we have protruding tongue out of it. So this is tongue protruding. Then we have vertical poles called epicanthus on either side of the nose. Skin fold at eye corner. So eye corners have skin folds and epicanthus below. Then we have short neck, small flat hands with stubby little finger, loose jointedness at ankle, mental retardation, cardiac deformities, under deformities. So you can say that underdeveloped gonads and genitals. And risk of Down syndrome is higher in parents below 18. Fathers above 49 and mothers in age group of 35 to 40. It is caused by non-disjunction of 21st chromosome pair during NFA's first. As a result, N+1 gametes are formed. So, this N+1 gamete is formed. Fertilization with normal gamete N results in trisomy. So we have N plus that's N plus 1, or you can say 2N+1. That disorder does not run in family. So however, a rare familial Down syndrome also develops due to non-translocation of major part of chromosome 21st to chromosome 14. So this is another one. This is familial one. Normal incidence rate is 1 in 1500 births. So 1 in 1500 births rate of this syndrome. Death occurs in 50% of cases. In the first year, survivors have higher chance of catching leukemia and Alzheimer's disease, as said earlier. Now coming to the next one after this Down syndrome. So we have that second one, that is Edwards syndrome. It's actually trisomy of 18th chromosome. 18th chromosome gets trisomized, malformed ears, receding chin, defective nervous system. This is a problem with these people. We have third one also, that's called the Patau syndrome. This is a trisomy of 13th chromosome. Here we have hair lip and cleft palate problem. So then we have completed 11th or 12th, that is sex chromosome abnormalities. So we have sex chromosome abnormalities. In this sex chromosome abnormalities, we have first, and let us take it fresh. So we have, okay, so let's write it here. We have first, that is Turner syndrome. It's given by Turner et al. 1938. So there is a single X chromosome actually. So we have 44 plus XO due to fusion of 22 plus O ovum and with gynosperm 22 plus X. Incidence rate is 1 in 3,000 female births. That defect is also called female Turner syndrome because it happens most in females. So male Turner syndrome does not survive. 44 plus Y does not survive. That's why we have found only females. The individuals are females with limited secondary sex characters, rudimentary ovaries, lack of menstrual cycle. We have spare pubic hair, undeveloped breasts. Then we have short stature, webbed skin over neck, low set ears. We have shield-like chest. We have abnormal jaw formation, subnormal intelligence, etc. These are problem with these people who are having this. So mostly in females because males do not survive. So then in second we have Klinefelter syndrome. It's given by Klinefelter in 1942. It's a sex complement is XXY. Incidence rate is 1 in 850 births. So it's 1 in 850 births. It is produced by fusion and normal androsperm. This 22 plus Y with egg ovum to 2X. This 22 plus XX. Or normal egg 22 plus X with the sperm having both chromosomes 22 plus XY. The individuals are males with undeveloped testes, azoospermia, occurrence of bare bodies, feminine secondary sex characters. That's what's called a gynecomastia. So they develop, kind of milk producing system, but are males. And like very few body hair and enlarged breast, small prostate gland, long limbs with occasional mental retardation. So gynecomastia. Klinefelter syndrome with XXXY and XXXXY is also known. So we have these individuals with this problem of having extra X. So we can have XXXY or other. Then we have third super female. Sex complement is XXX, XXXX or 5 times X or triple X syndrome is more common. They are mentally retarded and commonly infertile. Occurrence is 1 in 1200 female births. So we have 1 1200 female births. Now taking another fourth, that is super males, Jacobus syndrome. The sex complement is XYY. That's Hoschika has developed it in 1962. Incidence rate is 1 in 1000 male births. So we have exactly 1 in 1000 male births. And there is, overproduction of testosterone and defective producer